Okay, next topic, Lac Operon. Now, before starting Lac Operon, I would like to discuss what exactly is Operon. Operon is a group or it is a cluster of a gene with related function. Any group of a gene, any set of a gene or any cluster of a gene together with some related function is generally called as operon. So operon is a system of a gene, you can say, right? So you know very well I have written Lac Operon. Lac stands for lactose operon. Everyone knows what is lactose. Lactose is a type of a sugar. It's a kind of a disaccharide. Okay, so here we come across Lac Operon. Lac Operon is a operating system. Okay, so we all know that glucose is the most stable form of the energy required by the body, right? It is an instant source of an energy, but in absence of glucose, body utilizes another source of energy which is called lactose, right? Okay, so when I talk about the lactose, either the lactose may be present or either the lactose may not be present, right? That is, they have two conditions: if the lactose is available or if the lactose is unavailable. Okay, so let's talk about Lac Operon. If lactose is unavailable, right? Okay, if lactose is unavailable or if lactose is available, there are two major components which are called promoter and operator on a gene, right? What is promoter? We have discussed. Promoter is a sequence of a DNA where the RNA polymerase enzyme comes and bind, correct? The process where the RNA polymerase enzyme comes and binds, it's generally called as promoter. So a sequence of a DNA where RNA polymerase enzyme comes and binds is called promoter, and second is called operator. Operator is a sequence of a DNA where the repressor protein comes and bind, right? So where the repressor protein comes and bind is called operator, and where the RNA polymerase enzyme comes and binds is called promoter, right? So two conditions: if lactose is unavailable or if the lactose is available. First, if lactose is unavailable, that means if the lactose is not present. If lactose is unavailable, repressor messenger RNA produces repressor protein. This repressor protein will go and bind to the operator, right? Once the repressor protein goes and binds to the operator, now the RNA polymerase enzyme cannot bind to the promoter, right? I repeat it. If lactose is unavailable, repressor messenger RNA produces repressor protein. Repressor protein molecule goes and binds to the operator. Once the repressor protein goes and binds to the operator, now this repressor protein will block the RNA polymerase to bind at the promoter. Take a very simple example. If we want to block anyone on WhatsApp, right? Immediately, whatever the contact is there, we go and we block it. Now, once the contact is blocked, neither you cannot send the message, neither the person cannot send you the message, right? Like that, here the RNA polymerase enzyme is been blocked, which cannot be bind to the promoter, and thus the transcription generally stops, right? Okay. Now, after some period of time, what will happen? The lactose will increase. That means the lactose is available, right? So, if the lactose is available, then lactose itself acts as an inducer. Remember one thing, if the lactose is available, then lactose will itself act as an inducer. Inducer is nothing but it is going to carry forward the process of initiation, right? Now, lactose will act as an inducer. It will remove the repressor from the operator, right? It will remove the repressor from the operator and will bind itself with the repressor to form repressor inducer complex. Right? Okay. Now, once the repressor inducer complex is formed, operator is now totally free. Now the RNA polymerase enzyme can come. Now the RNA polymerase enzyme can come and bind to the promoter. Why now the RNA polymerase enzyme can come and bind to the promoter? Because there is no repressor protein on the operator. Hence, now the operator is free. Now the RNA polymerase enzyme can come and it can bind to the promoter. Now, First, this RNA polymerase enzyme will come and encounter Z gene, right? Now, this Z, Y, A are the structural gene which are present, right? Now, this RNA polymerase enzyme will come and first encounter the Z gene. The Z gene produces an enzyme called beta galactosidase, right? Beta galactosidase is the enzyme which will break the bond between glucose and galactose to form lactose, right? Okay, glucose and galactose both are your monosaccharide. In between them, the bond will be broken down to release the sugar which is called lactose. And that is the function of beta galactosidase, right? Now, from the Z gene, the RNA polymerase enzyme will go and encounter the Y gene. Y gene produces the enzyme called permease, right? Now, what is the function of the permease? Permease will carry the lactose to the cell, right? Now, whatever the lactose which has been formed, that lactose will be carried forward with the help of the enzyme permease to the cell. That's why permease is called lactose carrier, as it transports the lactose to the cell. From the Y gene, the RNA polymerase enzyme comes forward to the A gene. A gene produces the enzyme trans-acetylase. Trans-acetylase is the enzyme which will require for the transfer of your acetyl group, right? Now, what happens is that I said that the permease is the enzyme which generally carries the lactose to the cell. That's why it is called lactose carrier. Now, when the lactose will be available to the cell will utilize the entire lactose. I mean to say, the cell will use the lactose. And just think, if the lactose is utilized by the cell, then the amount of the lactose will get decreased, right? After a certain period of a time, again the lactose will decrease as the cell has utilized the lactose, right? It means that the lactose is unavailable. Correct now? Lactose is utilized by the cell. If the cell uses the lactose, the proportion or the amount of lactose will decrease. Decrease means again the lactose is unavailable. So again, if the lactose is unavailable, repressor messenger RNA will produce the repressor protein. Repressor protein will go and bind to the operator. Operator will block the RNA polymerase to bind at the promoter. Thus the transcription stops. After a certain period of time, again the lactose will increase. Again, the lactose will act as the inducer. Lactose will remove the repressor from the operator and itself bind with the repressor to form repressor-inducer complex. Again, now operator is free, so RNA polymerase enzyme will come and bind to the promoter. RNA polymerase will jump to the Z gene. Z gene will produce beta galactosidase enzyme, which will break the bond between glucose and galactose to form lactose. Right? From the Z, it will move to the Y gene. Y gene will produce permease enzyme. Permease will carry the lactose to the cell, so called as lactose carrier. From the Y gene, again it will jump to the A gene. A gene will produce the enzyme trans-acetylase, and trans-acetylase will transfer the acetyl group. Right? It means that the cell is going to utilize the lactose. Again, the lactose will decrease. Again, the lactose is unavailable. Again, repressor will go and bind to the operator and will stop the process of transcription. This switch on and off mechanism is called Lac Operon. So, in this way, the Lac Operon system regulates in our body. Thank you.